Now we are moving on to Chapter 12, Part 3. So we're going to talk about what happens at the synapse of our neurons. So the synapse is that specialized site where the neuron is communicating with another cell. It's the site of communication. Remember, we have that presynaptic neuron, which is the one that's sending the message. That action potential is traveling down to the synapse. And then we have the postsynaptic neuron or cell, depending on our situation, that's receiving the message. We have two different types of synapses in the body. We have electrical synapse and chemical synapse. So an electrical synapse is less common, and it involves direct physical contact between two membranes. So the cells are actually touching each other, up against each other, and they're locked together thanks to gap junctions. Remember those gap junctions that we talked about at the beginning of the semester? And we can see them here in this picture. The gap junctions were those ones that kind of made a little tunnel between the cells. They had those little connexins that kind of joined together into a little cylinder with a pore in the middle. And remember, the purpose of those junctions was to allow ions to pass between the cells via those pores in the gap junction. So what happens in electrical synapses is that the local current that's created by the action potential is going to affect both the cells. And the action potential is going to propagate to the second cell because they're actually in contact with each other. Because of this contact, it provides a very quick and efficient form of message transmission. And the places that we find these electrical synapses are some areas of the brain and the eye and the ciliary ganglia. Remember, ganglia are cell bodies, groups of cell bodies in the autonomic nervous system, and so ciliary ganglia are involved in the eye. The chemical synapses are more common, and they're what we're going to spend the most time talking about. With the chemical synapse, the signal is transmitted across a gap between the two membranes, thanks to neurotransmitters, which are chemical substances. And this is the most common type of synapse we find between two neurons, and the only type of synapse we find between neurons and other cells, such as neuromuscular junctions, where a neuron is communicating with the muscle cell, neuroglandular junctions, where a neuron is communicating with the gland. So those two cells are separated by that space, and that space is called the synaptic cleft. And we can see that in this image here. So the neurons, I'm so bad at knowing where this camera is. So the neurons are in close proximity, but not actually touching. There is a gap between the two, and the neurotransmitter has to cross that gap and reach that second neuron for it to receive the message. And once again, we have the presynaptic cell or neuron sending the message, and then the postsynaptic cell or neuron receiving the message. At the axon terminal, remember when we have our neuron, I'm going to draw a little very basic neuron here. Oops. So we've got our cell body here, our axon, our telodendria. And remember, those end in, we can call it the axon terminal, the synaptic bulb, the synaptic terminal. So at that terminal, at that end of the telodendria, and that's what we're seeing here in this image, we have mitochondria and then thousands of little vesicles that are full of those neurotransmitters. And what happens when that action potential travels down to the axon terminal, that axon terminal is going to flood that synaptic cleft, that space between the two cells, between the two neurons, with neurotransmitters. And then those neurotransmitters are going to bind to a receptor on the postsynaptic membrane. So those neurotransmitters are released by the presynaptic neuron. They're going to cross the cleft and then bind to a receptor on the postsynaptic membrane. And changes in membrane permeability produce, because of the neurotransmitter binds that receptor on the postsynaptic cell. Remember, we have cells that we have receptors on membranes that bind chemicals or ligands. Those are our chemically or ligand-gated channels. And when those channels bind the ligand, which in this case is the neurotransmitter, that channel opens and allows things like sodium ions to flow inside. So that binding of the neurotransmitter is going to change the permeability of that postsynaptic membrane to things like sodium or potassium. That binding that opens the channel and changes membrane permeability is going to produce a graded potential. Remember, a graded potential is a localized change in the membrane potential thanks to ions coming into or leaving the cell. And so in the case, for example, of a ligand-gated sodium channel, the ligand, the neurotransmitter, would bind the receptor. It would open the channel, open the gate, allow sodium to flow into the cell. Sodium has a positive charge, and that would cause a depolarization, a localized depolarization, where the membrane potential is changing to a less negative value. If that graded potential is large enough, then we can generate an action potential in the postsynaptic cell. Remember, for an action potential to be generated, we have to reach a certain level of depolarization at the trigger zone. If we depolarize to that threshold level at the trigger zone, then the action potential will be generated in that postsynaptic cell. And whether or not that happens is going to depend on the amount of neurotransmitter that has been released by the presynaptic cell, the sensitivity of the postsynaptic cell. So if we only have a small amount of neurotransmitter being released by that presynaptic cell, we're not going to be opening as many channels. We're not going to be getting as great of a depolarization on that postsynaptic cell. That postsynaptic cell also may have a higher or lower sensitivity to the neurotransmitters that are arriving that may affect how great of a depolarization we have from that graded potential. So after all of this happens, the axon terminal from that presynaptic neuron is going to reabsorb and reassemble particles of the broken down neurotransmitter that are left at the synapse. And we have a continuous supply of the neurotransmitters being made by the cell body. So I'll redraw my neuron here. So here's our cell body, our telodendria ending in our axon terminal. So our neurotransmitters are continuously being made by the cell body, and then that anterograde flow is going to carry them down the axon to the axon terminal, where then they can be released. And after they're released, once we're done using them, they're going to be broken down and taken up again into the axon terminal, where they may be recycled. So cholinergic synapses are a specialized synapse that release acetylcholine as our neurotransmitter. And we find these cholinergic synapses at all neuromuscular junctions involving skeletal muscle fibers. So any junction where a neuron is communicating with a skeletal muscle fiber is going to be a cholinergic synapse, where acetylcholine is the neurotransmitter involved. We also find this in many of the synapses in the central nervous system and all neuron-to-neuron synapses in the peripheral nervous system, and then all neuromuscular and neuroglandular junctions in the parasympathetic division of the autonomic nervous system. So this is a very common type of chemical synapse, this cholinergic synapse, which involves the neurotransmitter as acetylcholine. So what happens at the cholinergic synapse? That action potential Draw my neuron here. So that action potential is going to travel down our axon and arrive at the axon terminal. And remember that action potential is a depolarization that's traveling down that membrane. It's an excitable membrane, and so that depolarization, once it starts, travels all the way to the end, to the axon terminal. And that depolarization is actually going to trigger some voltage-gated channels in the axon terminal. If you remember from last lecture, the axon terminal has these calcium-gated voltage channels. Remember, for a voltage-gated channel to open, it requires a change in the membrane potential to open it. That local current that's traveling down is going to trigger the opening of a voltage-gated channel. So once that action potential comes down and depolarizes the axon terminal, it triggers the opening of those calcium channels that are voltage gated in the axon terminal. What happens then is that calcium that's outside the cell, so extracellular calcium ions, are going to actually come into the axon terminal, and that triggers exocytosis of acetylcholine. Remember, our neurotransmitters are stored in vesicles inside that axon terminal. So we've got, I'm going to erase this real quick and draw some more. Oops. So this is going to be a close-up of our axon terminal. So the very end of our telodendria. And we're going to have these vesicles full of acetylcholine, ACH. It's really hard for me to draw small. Okay, and then we're going to have these, whoops. No, I didn't want to delete that whole thing. We're going to have these gated channels, and we're going to have calcium hanging out outside here, extracellular calcium. So that action potential, that depolarization, is going to travel all the way down to the axon terminal, and that depolarization is going to trigger this gate to open. That's going to trigger calcium to come into the cell, and that's going to trigger exocytosis of acetylcholine out of the axon terminal. So it's kind of a multi-step process to release that acetylcholine from the axon terminal. The acetylcholine is then going to cross the synaptic cleft to the postsynaptic membrane, and there are receptors on the postsynaptic membrane that bind acetylcholine. So they're chemically or ligand gated receptors. So here's our postsynaptic cell, and we've got these special chemically gated channels. So the acetylcholine is going to come in and bind that chemically gated channel, and that's going to open it and allow influx of sodium into that postsynaptic membrane. And what does influx of sodium cause? Remember, sodium has a positive charge coming into the cell, causes a depolarization. That membrane potential is going to go to a less negative value. It's going to depolarize it and create one of those graded potentials. The graded potential is that localized change in membrane potential. And then depending on how large that depolarization is, how large that graded potential is, we may or may not trigger an action potential in that postsynaptic cell. That acetylcholine that bound to the postsynaptic membrane receptor is then going to be removed from the synaptic cleft by acetylcholinesterase, or ACHE. And the acetylcholinesterase is going to break acetylcholine into acetate and choline. If that graded potential is big enough, sorry, I already said that to you. And so the acetate and choline are going to be basically recycled by the presynaptic cell or the body to create more acetylcholine. So these are images from your book just going through these steps. So here we see an action potential is traveling down the axon to the axon terminal. This is the axon terminal that we're seeing here. That action potential travels down to the axon terminal. Next step is these voltage-gated calcium channels are going to open in response to that action potential, that depolarization, reaching them because they're voltage-gated calcium channels. Then that extracellular calcium is going to flow into the cell, and then the vesicles of acetylcholine that have been waiting in the axon terminal are going to fuse with the membrane and cause exocytosis of our acetylcholine into the synaptic cleft. On this is our postsynaptic membrane here. So that acetylcholine crosses the synaptic cleft, and it's going to bind to these chemically or ligand-gated channels on the postsynaptic cleft. And that's going to trigger opening of those channels, and then sodium is going to flow in. Sodium is going to initiate a graded potential or an action potential depending on if threshold is reached. So if we depolarize to threshold, then we will trigger an action potential. And then that acetylcholine is going to get removed from that ligand-gated channel by an enzyme called acetylcholine esterase. The acetylcholine is then going to get broken into choline and acetate, and the choline is going to be taken back up by the axon terminal, and it's going to combine with something called acetyl-CoA to make more acetylcholine and then get stored in the vesicles again for future use. And then the acetate is going to be taken up by other cells or tissues in the body. So make sure that you know that removal of the acetylcholine from that postsynaptic membrane, from that ligand-gated channel, is going to be performed by acetylcholine esterase. So we can have what's called a synaptic delay, which is basically a delay of between 0.2 and 0.5 milliseconds between the time that an action potential arrives at the axon terminal and the effect happening on the postsynaptic membrane. So once again, my poor drawing. So here's our axon terminal. Here's our postsynaptic membrane that has the ligand-gated channels. So there's a delay from the time that the action potential reaches this axon terminal to when this postsynaptic membrane is going to respond. And most of this delay in time is due to the time that it requires calcium to flow into this the presynaptic cell. So remember, for the neurotransmitter to be released, we have to have calcium coming into the axon terminal. And then that neurotransmitter is also going to take time to cross that synaptic cleft. So both of those things take a little time, and that's what causes our synaptic delay. What this means is that the fewer synapses we have in a pathway, the faster the response is going to be. If we have only one synapse, then we're going to have a very fast response. And that is why important reflexes that might be needed for survival or preservation of our well-being are going to have only one synapse. We would have a sensory neuron synapsing directly on a motor neuron rather than traveling over several synapses, over several neurons to get the desired effect. So the fewer synapses we have, the less synaptic delay, the faster the response is going to be. We can also have what's called synaptic fatigue, and this can happen when those neurotransmitters aren't recycled quickly enough to meet the demands of an intense stimuli. So the response of the synapse is going to weaken until that acetylcholine gets replenished. So if we have an intense stimulus that is triggering repeated action potentials, we can run out of our acetylcholine, and then we have synaptic fatigue where we are not triggering that desired effect because we don't have enough acetylcholine to release across the synaptic cleft to bind to the postsynaptic cell. Other factors that can influence what effect the release of a neurotransmitter will have on that postsynaptic cell are going to include the availability of receptors on the postsynaptic cell. So if our axon terminal, here's our postsynaptic cell. If we've got lots of receptors for our ligand on that postsynaptic cell, it's going to be a different response than if we only had, say, one receptor for that specific ligand on the postsynaptic cell. Because if we have lots of gates that are opening, that's going to cause a greater depolarization as that sodium flows in than if we just had one gate opening. The frequency of neurotransmitter release is also going to have an effect on the postsynaptic cell. And then the number of neurotransmitters that are released. So if we have even if we have a bunch of receptors here to bind our neurotransmitter, if we only have one neurotransmitter that's released, the effect is not going to be as great. So we've discussed acetylcholine here, but we do have other neurotransmitters in our body that our neurons are continuously being exposed to. And we can classify these transmitters based on their activity into excitatory or inhibitory neurotransmitters. When we say an excitatory neurotransmitter, that neurotransmitter is going to result in a depolarization of the postsynaptic membrane, thereby promoting the generation of an action potential. So, draw my little drawing again. So, if the neurotransmitter released by this presynaptic cell binds and causes an influx of sodium, that would cause a depolarization of this membrane, which could generate an action potential. So that's why it's called excitatory. We're getting close to generating an action potential. We're working towards that. Whereas we can also have inhibitory neurotransmitters. And inhibitory neurotransmitters, where's my cursor? Inhibitory neurotransmitters are going to result in a hyperpolarization of the postsynaptic membrane, thereby suppressing an action potential. Remember, for an action potential to occur in that postsynaptic membrane, we need to depolarize to threshold. So we need to get to a less negative membrane potential. If we're getting a hyperpolarization, we're making that membrane potential more negative. So we're moving away from the ability to generate an action potential. So it's suppressing an action potential, and that's why it is called an inhibitory neurotransmitter. And so an example of what would happen there is if we had a neurotransmitter leaving here that was binding. I need more space to draw on this slide. If this neurotransmitter that was released and bind this gated channel, if this gated channel that it bound to was a potassium gated channel, then potassium is going to be exiting the cell. Those positive potassium are going to be exiting the cell, and that's going to cause a hyperpolarization of the postsynaptic membrane, so moving away from the action potential. The effects of the neurotransmitter on the postsynaptic membrane isn't always black and white though, because the effects can depend on the properties of the receptor, not the nature of the neurotransmitter. So for example, in heart muscle, in cardiac muscle, the release of acetylcholine at the neuromuscular junction has an inhibitory effect. So if we bind acetylcholine to the receptors on the cardiac muscle cell, we get a hyperpolarization of that cardiac muscle membrane. But in skeletal muscle, we have the opposite. When we bind acetylcholine, we get an excitatory effect because the binding of acetylcholine causes a depolarization of the skeletal muscle membrane. So what happens when the neurotransmitter binds to that membrane can depend on the receptor itself, not necessarily the neurotransmitter. So one neurotransmitter may cause different effects in different parts of the body. It might be inhibitory in one place and excitatory in another. Neurotransmitters can have a direct effect on membrane potential. That's kind of what we've been talking about here previously. So acetylcholine could bind a receptor, trigger opening of that sodium gated channel, and then sodium could flow in and directly cause a depolarization of the membrane. That's called an ionotropic effect. Or we can have indirect effects. So that first neurotransmitter could bind a receptor on the membrane in the postsynaptic cell, and that could end up resulting in a series of secondary effects that then result in the opening of a membrane channel. So it could be that that binding of the receptor isn't directly causing a depolarization, but it's triggering a series of events that will cause a depolarization down the line. We have different classes of neurotransmitters. These include biogenic amines, amino acids, neuropeptides, and dissolved gases. Let's take a look at some of the more important neurotransmitters found in our nervous system, and some of these will appear in the unit on the brain as well. Norepinephrine is sometimes abbreviated as NE and is also sometimes called noradrenaline. We find this neurotransmitter in the brain and portions of the autonomic nervous system, and we find this at what we call adrenergic synapses. We mentioned cholinergic synapses, which are found with acetylcholine. So these adrenergic synapses are found with noradrenaline or norepinephrine. And this neurotransmitter usually has an excitatory effect on the postsynaptic membrane, and it's involved in consciousness and alertness, concentration abilities, energy, and body temperature regulation. Dopamine is a CNS neurotransmitter that we find in the brain, and this one can be either excitatory or inhibitory, depending on the location. Dopamine is involved in our motivational drive of our brain, what makes us want to do things, and so it is related to pleasure. Obviously, if something is pleasurable to us, we want to do it again. It's also involved in our emotions and plays a role in addiction, and is involved in subconscious motor function, so subconscious control of our muscles. An example of the inhibitory effect of dopamine: in one part of our brain, the release of dopamine prevents the overstimulation of neurons that control skeletal muscle tone. So it has an inhibitory effect in terms of controlling skeletal muscle tone. If we get destruction of the neurons that produce dopamine in this area, we end up getting this rigidity or stiffness of muscles, which is seen in patients with Parkinson's disease. The drug cocaine inhibits the removal of dopamine from synapses in specific areas of the brain. And because that dopamine is not being removed as a result of the drug cocaine, that rise in dopamine concentration at the synapses, it is what results in what we call the high or pleasurable effect experienced with that drug. Serotonin is another CNS neurotransmitter. It has an effect on our attention, emotional states, our appetite and body temperature, and is also involved in our sleep and wake cycles, thanks to interactions with some other neurotransmitters. if we do not have high enough levels of serotonin being produced, we can end up with effects on attention and can also end up with depression. You may have heard of some drugs, such as Prozac or Zoloft, being described as SSRIs, and this stands for selective serotonin reuptake inhibitor. And what they do is they stop the reabsorption of serotonin by the axon terminal, so that serotonin is not being taken back up into the presynaptic cell, the presynaptic neuron. So we end up getting an increased serotonin concentration at the synapse, which can help relieve some of those symptoms of depression by making that serotonin more available. And then lastly, we have gamma aminobutyric acid, or GABA. And this neurotransmitter generally has an inhibitory effect. We don't completely understand its function, but it is found in 20% of the synapses in our brain, and we believe it's related to anxiety because some anti-anxiety medications work by enhancing the effects of GABA. Neuromodulators are basically substances that are going to alter the response of the postsynaptic neuron to the neurotransmitter, and they can work in various ways. Some examples would be neuropeptides. These are made and released by the axon terminal, and they may alter the rate of the neurotransmitter release, or they may change the response of the postsynaptic neuron to a neurotransmitter. Opioids are another example. You may have heard of opioids before because they are similar in structure to some drugs such as opium and morphine. Sometimes if a drug is close enough in structure to a naturally occurring substance, then we can have binding of the drug to the receptor for that naturally occurring substance. So in our body, we do have opioids that are naturally occurring, and we have three different classes of opioids. We have enkephalins, endorphins, and dynorphins, and the dynorphins are the most powerful. And these opioids naturally can provide pain relief, just like the drugs, opium and morphine, can provide pain relief. And the way they do this is they are neuromodulators. So they are going to act by inhibiting the release of the neurotransmitter substance P. Substance P is a neurotransmitter involved in pain, especially visceral pain, so pain of the internal organs. And so the opioids are going to inhibit the release of that neurotransmitter, which causes pain, and that's how they are going to decrease the level of pain that you're feeling. So how does our neuron process all this information it's receiving? What is it going to do with it? We can have one neuron that is receiving both excitatory neurotransmitters as well as inhibitory neurotransmitters at the same time. So how does it respond? Well, the net effect on the membrane potential of the cell body, the axon hillock in particular, right, is going to determine if an action potential is produced at that neuron. This combination of adding these potentials together that are coming into the neuron is called summation. Basically, with summation, we're adding all these membrane potential changes together. The membrane potential changes may be coming in repeatedly at the same spot, or they might be coming in from different locations on that neuron. And if we add these together, that summation of these potentials, if it leads to a depolarization that's big enough to reach threshold at the trigger zone, that axon hillock, then an action potential is propagated down the axon. So our graded potentials develop in a postsynaptic cell membrane in response to the neurotransmitters that bind it. We have two different types of postsynaptic potentials. So when I'm saying postsynaptic potential, it's that graded potential produced by the postsynaptic cell. So here's our first neuron, and then here's our second neuron. So here, this is our postsynaptic cell, right? It's got some sort of receptor. The presynaptic cell is going to release some neurotransmitters that are going to bind that receptor. That's going to open a channel in most instances, causing either a depolarization or a hyperpolarization depending on what type of channel it is. Are we letting sodium in? Are we exiting potassium? But whatever happens, we are going to be producing a graded potential there. The graded potential, remember, is that localized response. So if we have, so that graded potential at that postsynaptic cell is called a postsynaptic potential because it's taking place at the postsynaptic cell. So we have two different types of postsynaptic potentials we can have. We can have an excitatory postsynaptic potential, and with an excitatory postsynaptic, these are tongue twisters, an excitatory postsynaptic potential, we've got a graded depolarization of the postsynaptic membrane. Remember, to trigger an action potential, we need to depolarize that membrane to the threshold level. So it would make sense that if it's an excitatory postsynaptic potential, we would be depolarizing. Conversely, so an example of that would be the binding of acetylcholine at our cholinergic receptors. Acetylcholine binds that receptor on the postsynaptic cell that opens that gated sodium channel, and that leads sodium enters, and that leads to a depolarization of that membrane. Conversely, we have inhibitory postsynaptic potentials. With an inhibitory postsynaptic potential, we get a graded hyperpolarization of the postsynaptic membrane. So remember, hyperpolarization is moving to a more negative value. So we're moving away from that threshold that we're trying to reach to generate an action potential. An example of this would be opening of those chemically gated potassium channels. So remember, when those potassium channels open, potassium exits the cell. That positive potassium exiting causes a hyperpolarization. It causes the membrane to have a more negative value, a more negative potential. So we're moving away from our goal of generating an action potential, which is why it's called an inhibitory postsynaptic potential. While hyperpolarization is continuing, the neuron is what we call inhibited, which means in order to depolarize that neuron to threshold to generate an action potential, we would need a larger than usual stimulus because we are hyperpolarized. So it's going to be we're going to need a greater change to reach threshold. So that's called inhibition of the neuron. To trigger an action potential, often one excitatory postsynaptic potential is not enough. So sometimes we need more than one excitatory postsynaptic potential. And our excitatory postsynaptic potentials and inhibitory postsynaptic potentials can all be happening at the same time or one after the other on the postsynaptic membrane. And they basically combine together through a process called summation. All right, just a quick note about drugs. This little GIF or GIF, I can't ever remember which it is from a great movie called Almost Famous. It probably came out when I was in high school. It's a very good movie if you haven't seen it. So how do drugs work? Drugs can work in different ways, and this little illustration just kind of lists some of the different ways that drugs can work. So in a normal, when we're not looking at drugs, a normal sequence of events across a synapse, we have a neurotransmitter, right, that's released by the presynaptic cell, binds a receptor on the postsynaptic cell membrane that triggers a natural effect, be it a depolarization of the membrane because sodium is flowing in. So that's what normally would happen. Some of the effects of drugs, we could have, in this instance, a drug may increase how many neurotransmitters are released, and that increases the response of the neuron that's receiving it. So So if we have a whole bunch more neurotransmitters released as a result of that drug, then we have more of those receptors getting bound, and so more of those channels opening and depolarizing, so we can have a faster effect. We can also have instances where a drug may decrease the number of neurotransmitter release, and that decreases the response of the postsynaptic cell or postsynaptic neuron. So we might have less of an effect at that postsynaptic cell than we normally would. So we might not be producing an action potential when we normally would be because we have less neurotransmitter because of that drug. We can also have instances where the drug might fit into the receptor for a natural neurotransmitter and cause a similar response. So the drug might look close enough to the natural neurotransmitter in our body that it's able to bind that receptor and open that channel and produce the same effect that it would if it was a natural neurotransmitter. And that's kind of what we saw when we were talking about opium and morphine, those are drugs that provide pain relief because they are similar to those natural endorphins. Then we can also have psychoactive drugs that fit into the receptor but prevent it from opening. They fit into the receptor, but they don't activate that channel. They're different enough that, or their action is such that that channel does not open. So they're not opening that channel to produce that natural effect. And not only that, but they're taking up the space on that receptor, so our naturally occurring neurotransmitter cannot attach and perform its natural action. So those are all different ways in which drugs can cause an effect on the postsynaptic cell. So drugs can release 2 to 10 times the amount of dopamine that natural rewards do. So when I say natural rewards, I mean something like giving your kid a hug or petting your dog. So drugs release much more dopamine than a natural reward does, and they do it more quickly and more reliably. And as a result, when we take drugs regularly, our brain is going to make less dopamine naturally or eliminate some of those dopamine receptors because our brain thinks we don't need that dopamine anymore to be produced naturally. Some other drugs, alcohol's effects. Alcohol is going to increase GABA. Remember, we said GABA might be related to anxiety, and drugs that increase levels of GABA seem to work as anti-anxiety drugs. So this could be why some people are driven when they have stress to alcohol, because it can decrease the anxiety. GABA is also going to decrease glutamate, which leads to an increase in dopamine, which also leads to an increase in our pleasure drives. Cocaine is going to inhibit removal of dopamine from the synapses, so that dopamine is going to stay attached to its receptor, and that gives us this continuous high effect. THC is the chemical that's found in marijuana, and it's going to stimulate the release of dopamine. This can lead to euphoria, drowsiness, and an increase in appetite. Ecstasy is going to target our serotonin receptors, so it's going to affect our mood and can also impair cognitive function. Methamphetamine is going to affect dopamine, serotonin, and norepinephrine, and side effects of this drug could include anxiety, impulse control problems, irritability, and aggression. And then speed or amphetamines can cause excessive dopamine production and we can end up with people having schizophrenic-like behavior. Just a quick note about Parkinson's disease. We mentioned it before, and with Parkinson's disease, we get damage or degeneration of our neurons that produce dopamine naturally. If you remember back to the slide where we talked about this before, in some areas of the brain, dopamine actually has an inhibitory effect, and it prevents an overstimulation of neurons that control that skeletal muscle tone. So if that if there is damage and we're not getting that normal release of dopamine in that area, then we get overstimulation of skeletal muscle. And that's what gives us the rigidity or stiffness of the muscle seen in patients with Parkinson's disease. And that can be why it can be very difficult. They have tremors, and it can be very difficult for them to do fine-tuned movements such as holding a spoon and feeding themselves. Some other symptoms that we can see with Parkinson's disease include problems with balance and walking, rigid or stiff muscles, trouble swallowing, drooling, slow, quiet speech, and tremors. And then this is just a note about an interesting toxin that relates to what we've been talking about. I've mentioned this podcast before that I really like. It's called This Podcast Will Kill You, and they have a whole episode about this toxin. It's called tetrodotoxin, and you may have heard of it before because it's the toxin that's found in fugu, which is like a delicacy from this puffer fish. The puffer fish, I guess, produces this toxin. I think it might be in its liver or something, and so you have to have a very highly trained chef prepare fugu, or else you could end up getting too much of this toxin in your food, which can actually be fatal. So what tetrodotoxin does is it binds those voltage-gated sodium channels in the neuron. So remember, I'm going to draw our little neuron here. We've got our neuron. So we have our chemically or ligand-gated channels on the dendrites in the cell body, right, where we have to bind that ligand and sodium can enter. And then on the axon, we have voltage-gated sodium and potassium channels. And we need those voltage-gated sodium channels to be able to open in order for that action potential to flow down, to move down the axon. And so what tetrodotoxin does is it actually prevents those, it binds those channels and basically blocks the sodium from being able to enter. And so an action potential cannot be generated. You can, just think about all the problems you could have in your body if you are not able to generate action potentials in your neurons. Basically, your nervous system is not functioning. So symptoms of tetrodotoxin toxicity include a loss of sensation and paralysis of voluntary muscles, including the diaphragm and intercostal muscles. And eventually, you become unable to breathe. You are no longer able to move your diaphragm and intercostal muscles in order to breathe. Not all sodium channels bind tetrodotoxin. So when we're talking in this unit, we're kind of talking in general about different channels, but we have lots of different kind of specialized sodium channels and potassium channels in different parts of our body. So they're not all completely identical. And so not all of our sodium channels bind tetrodotoxin. There are specific ones that do. And some animals have more of the type that do not bind and therefore are able to consume species with tetrodotoxin. For example, I think it was in this episode of the podcast, they talked about the fact that there are some newts or salamanders in California, perhaps, that are not, actually no, what it was, I think it was a type of snake that did not have sodium channels that bind tetrodotoxin, and that made these snakes able to ingest either frogs or salamanders that produce tetrodotoxin. A lot of amphibians produce toxins naturally as a defense mechanism, and these snakes are basically immune to the tetrodotoxin, that if you or I ingested this frog or salamander would poison us. These snakes are immune to it and so are able to ingest these species with tetrodotoxin. And I believe that is the end of this lecture, but I think we will talk a little bit more about tetrodotoxin if we meet in person.